the mic to Dr. Ritu Trivedi to brief about the journey of this project. Please, ma'am.
Thank you, Sanjeev. And it is our pleasure that we are going to speak about our product Reunion as part of 1 week, 1 lab. And this product is Reunion. And as you can see on your screens, the product is Delbargia Sisu, and this product prevents bone breakdown and stimulates new bone formation, and it reduces bone turnover markers, and that is why this has been put in the category of for fast fracture healing and osteoporosis management. As much as I present this work today on my behalf, but there is a whole team of people who have worked at the back of it, who are responsible for bringing Reunion into the market. And you will see in this slide the research team below, from chemistry to biologists, to people from pharmaceutics and kinetics division, to business development and patent, which have all worked together to bring this product into the market. And it's my pleasure that Dr. Moria is here with me, with whose help we have been able to bring this product. And from pharmaceutics and kinetics, we have Prabhat, who has helped us take the product forward. Okay. So, I mean, why did we at all want to work and bring this product for osteoporosis? And the reason is that if you see around the world, it is 1 in 3 women and 1 in 5 men over the age of 50 years who suffer from osteoporosis, and it's in the category of unmet medical needs. And WHO says that it is characterized by low bone mass, and the low bone mass happens because of microarchitectural deterioration of the bone tissue, and this results in enhanced bone fragility and increase in fracture risk. And this is how the bones, if you see the panel in my right, you'll see the normal bone, and you'll see the bone that is fragile because of microarchitectural deterioration. And with respect to BMD, if you go to a clinician and if your spine or proximal BMD is less than minus 2.5 standard deviations, then you come into the category of osteoporosis. In this regard, we have a lot of treatment options available, from bisphosphonates to SERMs to HRT, to drugs with complex mechanism of action, but most of them are fraught with so many side effects, because of which we are always looking for new therapeutic options. And in fact, the bone formation therapy is something which is more sought for as opposed to the bone resorptive therapy. And in this category, we have only one, which I show you over here as parathyroid hormone, which comes in the market by the name of teriparatide or Forteo. But the problem with this anabolic drug is that it is the last line of treatment, and it is injectable, it is costly, and that is why we researchers are looking for therapy that can have the other therapies that can add to bone formation as a part of treatment. In this regard, my collaboration with Dr. Rakesh Moria, who's sitting just next to me, we looked, he got Delbargia Sisu as one of the options, and when he looked into the chemodiversity of Delbargia Sisu, there are so many compounds that were isolated. You can see these beautiful ringed compounds that have been isolated from Delbargia Sisu. In fact, out of all these, we were very lucky to see this blue one in the middle, which is K024, or which is the cavionin glycopyrroside, which is a novel molecule that we identified from Delbargia Sisu. And this was actually an excitement for us. We went ahead with this novel molecule in our hands to file a patent for it, which later on also got granted for this application for fracture healing and postmenopausal osteoporosis. And the several molecules were identified on the polarity-based chemistry. And out of the isolated 16 compounds that you see over here, 4 of them turned out to be osteogenic. That means they had bone formation ability and the capacity to form more bone. And out of these 4 of them, if you see the last one, K024, this was present in the highest amount. That is, you see 37.59 milligrams per gram per 5% of the total extract. So this was really exciting for us that not only did we identify a novel compound, but it also had osteogenic ability. Then I'll not take you into the details of the experiments, but I'll show you highlighted experiments of how we could see that the bone is getting formed. We had animal models that are WHO approved models for fracture healing and also for postmenopausal osteoporosis. The above panel shows that at different concentrations, Delbergia sissu could form bone. In the lower panel, you can see it is in the postmenopausal women, that is the women in which the estrogen is cut down or the reproductive capacity ceases. There also it could form bone. Not only it could form bone, but we also assessed the what kind of bone is getting formed. That means that what is the bone strength? Is the bone good enough? The formed bone has good strength even if more bone is getting formed. And our data showed that at 50 and 100 milligrams per kg body weight, actually it could form good bone. This is data that shows to us that how bone can be formed dynamically. It means you can assess bone micrograms per micrometer squared per day, which is the bone formation rate and a mineral apposition rate, which showed that Delbergia had the, not only it was osteogenic in nature, but we could also see that it was effectively forming dynamically bone in our histomorphometric data. From many of these experiments together and the highlighted experiments that I showed you, we could inference from this that Delbergia sissu could improve bone parameters, and it could do so by dual mode of action because it involved both the prevention of bone loss and bone formation of new bone. And we published all this data along with the patent, but our interest was in this novel compound. We wanted to see the magic of this novel molecule. We wanted to see if this in itself was osteogenic because it was present in such high concentrations in the entire Delbergia sissu extract that was standardized. And in our preliminary experiments showed that yes, it was effective. We did comparisons with parathyroid hormone, as I told you in the beginning, which is the standard of care, and genistein and alendronate at the concentrations and doses that are recommended for human usage. And from this data, you see the overrectomized versus the overrectomized plus. CAFG is the abbreviation of the full length of the novel molecule that I showed you earlier. And this shows that it could actually form bone at lower concentrations of 1 and 5 milligrams per kg body weight. This was exciting to us. Not only this, it had the ability to form new bone. The data that I showed you, micrometers per day is the rate at which the bone was getting formed with this novel molecule. And importantly, it was not estrogenic because for osteoporosis, hormone replacement therapy was the therapy that came just after postmenopausal women turned out to have low bone mineral densities. But the problem is that estrogen or estradiol जो है has side effects. It's causes cancer to the uterus and the breast. Therefore, all the time for long-term therapies, for example, for osteoporosis, we want to see that if the compound is estrogenic or not estrogenic, and we could conclude from these experiments that CAFG was not estrogenic. Not only that, it has effects on the chondrocyte cells. That means the cells, the beginning cells that start forming bone. These are some of the genes that are involved for chondrocyte formation or the chondrocytes at the fractured site. This was the experiment done to see the bone regeneration happening at the fracture site. And if you see, all the controls were taking over here, and CAFG at 1 and 5 milligrams per kg body weight could form bone much earlier than normally it is done with, which is the normal process of fracture healing, that is as early as 11 days at 1 and 5 milligrams, and importantly at a lower dose of 1 milligram per kg body weight it could form bone. Then we wanted to see what is the mode of its action, what is the mechanism by which this novel molecule forms bone. And we found that it is the Wnt beta-catenin signaling pathway that is actually responsible for activating bone formation. And CAFG, if you see, it activates the Wnt3A signaling pathway and allows beta-catenin to be localized inside the nucleus. It activates these transcription factors TCF, LEF, and thus activates RUNX2, which is the bone forming transcription factor, and we see the osteogenic response. And on the panel in the right, you will see all the experiments that were done to show how CAFG stimulates the Wnt beta-catenin signaling pathway to form more bone. Not only this, it also activates the BMP, the bone morphogenetic proteins, which are also responsible for forming bones by activation or the phosphorylation of the SMADs, and these SMADs activate the MAP kinases that allows it to internalize into the nucleus, and this thus activates the osteogenic response in the bone. We further, the panels again on my right are showing all the experiments that were done to show what I just explained to you, the Wnt and the BMP2 signaling pathway. Along with this, we went ahead to see the cells that are responsible for causing the osteoclast activity or the bone resorption activity and remain in close proximity with the osteoblast cells. What is happening to these osteoclast cells when CAFG is given in the physiological conditions? And we found that while it stimulates osteoblast action, we found that it inhibits the TRAP, the trattate-resistant acid phosphatase, which is responsible for resorbing the bone and thus limits osteoclastogenesis. And that is one reason why we said that it has dual mode of action. You see gene expression, and you see in the above panel how osteoclast cells are getting decreased. These are multinucleated cells that eat away bone. And thus, by its dual mode of action, by stimulating the osteoblast cells on one hand to form more bone, and on the other hand inhibiting the osteoclast cells by keeping their activity low, CAFG or the novel molecule that we showed just increased bone formation. So the mechanism of action for CAFG is through the mesenchymal stem cells. It increases the modulates the mesenchymal stem cell responsiveness to BMP2, and it promotes osteoblast differentiation by the Wnt beta-catenin mechanism. Detailed toxicity studies were done after we showed that how it is acting on the bone cells, and the 90-day oral toxicity and the 28 toxicity showed no signs of toxicity. Along with it, we also found that up to 1,240 milligram per kg dose, delbajaciclib did not show any toxic effects. In fact, it did not also show any induce any gene mutations by base pair changes or frame shifts in the genome of strains used, and therefore it was found to be non-cytogenetic. After all these studies were done, these experiments together this technology was licensed, and on the right you see Dr. Lal Hingorani, who is the CEO of the Farmenza Herbals Private Limited. The entire technology was transferred to him, and this was done as a rapid fracture healing agent. A clinical trial was immediately done after this, and the data of this clinical trial showed that yes, delbajaciclib does form callus, as we showed you its effects on the chondrocyte cells. It reduces pain, edema, with no adverse reactions whatsoever, and is well tolerated in humans. This was really a very, very exciting data for us because it showed that it had no toxicity effects. This product is finally in the market now. It is by the name Reunion. While the technology has been taken by Farmenza Herbals Private Limited, it is being marketed by Iris Life Sciences, and the process of how this happened and how we've been taken it to the market has been put in the form of a documentary, which is also available, the link that you should see over here on the website, on YouTube actually. This technology of ours brought in lots of awards and recognitions, suggesting that Reunion, yes, started doing good in the market, and it added to our confidence of bringing Reunion in the market. It was well appreciated at various platforms where we presented and got us a lot of awards and recognitions. It is a pride moment when things go from the lab to the market, and when they are appreciated by the lay press. It's a sense of pride for the institute and for the organization to take something like this to the market. And if we look at the worth of Delbergia Sisu, what it did, we began this work in around 2009, and now I'm speaking over here when it is in the market. You see the number of publications which are in the public domain. Anybody can go through these published data of what we've done with Delbergia. We've like 10 PhD students have done their PhDs while doing this work. And if you look at the social statistics, a lot of tribal population is collecting because it's from the leaves, the renewable source. In the Madhya Pradesh forest area, not only helping in protecting the trees, but it has provided a lot of revenue also to these tribal population. And if you look at the marketing data, you'll find that it has reached quite an amount to the patients, and a lot of patients have benefited. And we have not heard of any reported adverse action of Delbergia Sisu. We are still doing experiments. Our collaborator in the Pharmaceutics and Kinetics Division, Dr. Prabhat Mishra, where we estimate the content of the novel marker and Reunion so that the product that is in the market maintains its quality as long as it is there in the market. We haven't stopped over here. Delbergia, after trial 1 in the fracture healing trial, another trial was done that was in the postmenopausal women, and it demonstrated anti-osteoporotic activity. We found reduction in the serum alkaline phosphatase, and we found that the bone mineral density of the standard deviation that I showed you in my first earlier presentations, where you have bone mineral density falling below minus 2.5 standard deviations, we found that the BMD of these postmenopausal women did not decline further, and it benefited in their musculoskeletal symptoms. Taking all this together with trial 1 and trial 2, we have licensed the agreement with Avita Biomix. This is a company that is based in USA, and we want to take it as a pharmaceutical drug as per the US FDA guidelines. And this is the agreement that has been signed with Avita to take it forward. So we can say that Reunion will be available for poor bone health in general and osteoporosis in particular, with severity, morbidity worldwide. Thank you. I think this is a very, very happy moment for us to take Delbergia into the market. I have with me Dr. Rakesh Maurya also. I think he would like to say something about this.
Osteoporosis is a serious public health problem. It is one of the major problems in our aging society. Osteoporosis results in the bone fracture in the older member of the population, especially in postmenopausal women. In this program, we have screened 35 Indian medicinal plants. Out of this, Delbergia Sisu is one of the plants. And we have done this in very systematic manner, that in every step, from collection of the plant material, preparation of the extract, fractionation, and isolation of the compound, isolated compounds characterized, and all these steps, in every step the activity was monitored. And from this plant we have isolated about 16 compounds. All these compounds were, as described by Dr. Ritu Trivedi, were evaluated, and out of this only 4 compounds which have shown osteogenic activity. And one of the novel compound which was present in a very major compound also showed enhanced osteoporotic activity. I think this is what I want to say.
I think Dr. Maurya has summarized of all what I talked about just now, and I think it is a very, very proud moment for us and for the Institute to bring Delbergia in the market. And I leave all of you with a message that all of you love your bones, and please break barriers and not your bones. Thank you. Thank you so much.
Oh.